let's do proteins. So there's proteins, there's polypeptides, there's peptides, sorry, there's oligopeptides, and there's amino acids. Okay, so ultimately what I'm saying here is we want to get proteins to amino acids. Usable energy, right? I mean, usable form. So, I'm going to rub this out. Start off. Okay, first up, you've eaten a meal that's high in protein. Okay, and what you've ingested is a protein. And a protein has two ends. A carboxyl end and the amine end. Amino end. Okay? Good. Now you've got your stomach. Your stomach has two main cells, chief, parietal. These act to secrete different things. Okay? So, what your chief cell secretes is pepsinogen. Remember, whenever I think of pep, I think proteins. Okay, so it acts on proteins. But gen means it's inactivated, right? It needs to be activated. So parietal cells release HCl, which activates the pepsinogen and activates that enzyme to turn into pepsin. Okay? And this helps to break up the protein. But also, the HCl, right, is an acid, right? H plus, H plus. This also acts to denature the protein. So not just does the HCl activate pepsinogen, it also acts directly, the H plus ions directly act on the protein to denature it. Permanently change its structure. Right? So it breaks it down. Okay? It's broken it down. Now, what we get to is CCK again. Remember CCK, enteroendocrine cells. Okay? So what happens is CCK triggers the pancreas to release enzymes. This is what? Exocrine function. Anytime the pancreas releases enzymes into a body system, right? Not the blood, it's exocrine function. This will be pancreatic enzymes. Endocrine is within the bloodstream. So what happens is it'll release enzymes. So it release four main enzymes. Trypsinogen, chymotrypsinogen, peroxidase, and procarboxypeptidase. And you're going to be wondering, okay, they have GEN, some of these, because they're inactive. We've got to activate them first. So we've got another cell on the small intestine in the mucosal lining that release something called enterokinase. Okay, kinase phosphorylation, right? Anything with kinase, it plays with the phosphate group, PO43 minus. Right? Hold on a second. Okay, sorry. So trypsinogen, chymotrypsin, these are all inactive. Okay, enterokinase acts to activate this trypsinogen. So it activates trypsinogen directly. And then trypsinogen goes and activates chymotrypsinogen, peroxidase, procarboxypeptidase. And now, these enzymes can act to further break down this into dipeptides. Right? And tripeptides. And we're not done yet. Okay? You've also got enzymes within the small intestine, called dipeptidase, aminopeptidase. Okay, and these finally act on dipeptides and tripeptides to break them up into amino acids. Finally. Right. However, I want to note some of it still remains as dipeptides. We got to do something about that. And tripeptides. Okay? So it's same as before, enterocyte. It's just the lining of the small intestine, right? So, what happens is, amino acids always go by secondary active transport. So, all the amino acids go through the SGLT. What's SGLT? Sodium glucose transporter. Right? It uses this and it does secondary active transport. Right? Into the enterocyte. However, I mentioned before that dipeptides and tripeptides are still outside. Okay, we'll fix that, right? So you also have a H plus pump that transports H plus into the enterocyte. What about, we could use that as also a secondary active transport. That's exactly what we do. So we get dipeptides and tripeptides into the enterocyte. Oh wait, we can only use amino acids. Well, there's peptidase inside the enterocyte that act on the dipeptides to convert them to amino acids. Okay, now we've got amino acids. And next to it, right, so we've got the blood. Okay, now we know there's the sodium potassium transporter right here, but it doesn't use that amino acids to get into the bloodstream. Amino acids is a different one. It's called the sodium amino acid transporter. And it's able to transport the amino acids into the bloodstream for usage. Okay. Quickly, let's move on to saccharides. Okay. So let's start off with, got polysaccharides. Polysaccharides become oligosaccharides. And then monosaccharides. Monosaccharides is the general thing we think of. Monosaccharides are stuff like glucose are monosaccharides, fructose is a monosaccharide, also galactose is a monosaccharide. Okay? There's the three monosaccharides we want. So the end result, we want to get this. That's the end result. Okay? But we start off, so when we eat something, we have glycogen, remember? Glycogen in our food, starch, and cellulose. Now, cellulose is a plant form of... It's a plant form of energy. So the plant is able to break it down because it has a particular enzyme. So cellulose, we can't absorb it. So we can't break it down. We don't have the correct enzymes. But we can break down glycogen and starch. Now the general structure goes like this. It's a 6-carbon ring. I want you to think about it. Glycogen is just glucose joined. And glucose is what? C6H12O6. Remember the 6 carbons? That's why it's a hexagon. Okay, so people know. And this is called 1,4Y. We count here, we start with 1. And then we go around, right? If this is 1, we label this 1. That's 2, that's 3, and that's 4. That's why we label it 1,4 glycosidic bond, because now we're going to label that the origin. 1, 2, 3, 4. 1, 2, 3, 4. That's why we call that 1,4. This is the 4th bond now on that carbon. So it's 1,4 glycosidic bond. And we have enzymes that can actively break that. We have, in our saliva, produced by our salivary glands, we have alpha amylase. And alpha amylase, as the name suggests, directly breaks this bond. Okay, it can only break 1,4 glycosidic bonds. However, we've got unbonds. Because it's branched glycogen, we have 1,6 glycosidic bonds. So we're going to need further enzymes down the track. Okay, but it acts to further break it down. Good. And now we're going down, it's going throughout our system, and we get to the small intestines. And in the small intestines, you have, right, remember CCK, exact same thing, acts on the pancreas. This is the exocrine function again. And it releases what? Alpha amylase and other enzymes. The enzymes are, there's 4. Maltase, sucrase, lactase, dextrinase. Okay, those are the 4 enzymes. Maltase breaks up maltose and maltriose. This breaks up sucrose. This breaks up lactose. This breaks up dextrin. So all these enzymes and the alpha amylase that acts to break the 1,4 glycosidic bond further, all right, continue to break it, acts to give us the 3 forms of monosaccharides we want: glucose and galactose and fructose, sorry. The 3 monosaccharides we want. Okay, we're good now. Now we're just going to get it into the bloodstream. Okay, general, we still got our enterocyte, mucosal lining of the small intestine, right? Cells. So, generally, we've got the SGL transporter, right, for the enterocyte, which is a sodium glucose, right? These travel through, they can travel through secondary active transport, but only galactose and glucose can travel through the SGLT. So you get glucose through this, and you can get galactose, but fructose cannot. So we use a GLUT5 transporter for fructose, sorry. Fructose comes in through the GLUT5 transporter. And then we have the blood, right? We've got to get all these into the blood. How do we do that? We have GLUT2 transporters. And now everything can go through the GLUT transporters. You've got fructose, you've got glucose, and you've got galactose. These are monosaccharides finally in the blood. They will be used as energy later on. Thank you.